So I'm actually sharing my screen. I'm sharing this particular model. This is one of our tutorial models, which is a membrane photobleaching model, because this is a case where we do actually have to specify where some things exist in space. And let me just show you the results first for a second, so you can sort of understand what we're doing. So it's stuck up under the things. So this is a model where we're photobleaching an area of the membrane. So we need to be able to specify exactly the region that we're going to photobleach. So I can bring up the surface and show you the fluorescent molecules. Okay, so the idea here is that we're going to bleach a region of space. Let's see if I can bleach it at 2 seconds. Yeah, one more. I need to get a little after 2 seconds. So we're bleaching a square in the middle of the membrane, the top and the bottom membrane, and then allowing diffusion to allow the molecule to fill in by diffusion, which what Les showed in his little kinetic discussion of diffusion early this morning. So we need to specify where we're going to have that reaction occur. And the way we did it in this particular case, there's a number of ways to do this. The way we did it in this particular case was to define a species actually in the reaction that we're going to call laser. Now, laser isn't really a species, but it's just something that we can use to modify the reaction, to turn on the reaction. And with a species, we can put it in a particular space, right? So if we go to the specifications for that, then our laser we positioned in space, and actually we positioned it both in space and we've also positioned it in time. So we bleach for a certain period of time using Booleans to explain the initial condition. Okay, so virtual cell will allow Booleans to be put in to define functions. And everywhere the Boolean is true, it evaluates to 1, and everywhere the Boolean is false, it evaluates to 0. So in this case, spatially, we've said that everywhere that X is greater than 22 or less than 32, and everywhere that Y is greater than 35 or less than 45, it will evaluate to 1. So the value of that species will be 1 and the reaction will be turned on. Okay. And this will only happen at T, in this case, greater than 2 and less than 3. Now, this model, we just were doing a one-off, so we just hard-coded it with the exact positions and the exact times. But I model a lot of photobleaching reactions, and actually what it turns out that you don't always do it the same way. You can bleach bigger or smaller regions, and you can change the timing depending on how much you want to get bleached. So it's actually more convenient if you're modeling, doing things like this in the future, to have a start time and an end time parameter value, so you can change them as you're doing different simulations very easily, rather than having to go back and change the initial conditions every time you do that. So parameter values you can change in the simulation results. So this is good if you might create a parameter called ST for start and EN for end, and then you can you can vary those parameters as that changes with a particular experiment. And same for positions in space. So you can have, A be the one X position and Y be another X position, for example. It just makes it a little bit more flexible in the long run. The other thing I'll point out about, so this allows you to put things in any particular place in space, right, by using these types of Booleans. Or you can use other functions. You can use sign functions if you want it to vary in a regular fashion, for example. There's lots of ways you can just use that as an initial condition. I wanted to point out something else that's kind of a neat trick, which is how you would view those results in virtual cell. So if you go back to the simulation results, it's hard to put a position right on that membrane to show the results. So if I try to find where the top membrane is, let's go up there. I want to put see a plot of how that's changing. That's hard to do on the membrane because it's in 3 dimensions, so it's hard to figure out where you are, right? So I can build that plot. I won't take the time. Okay, but it's not exactly right. I want to see the whole bleach region because that's what I'm going to measure in my actual cell. So in fact, we have a little nifty function here called snapshot ROI. And almost nobody but me and its creator, Frank Morgan, knows how to use it, but it's really, really useful. So I'm going to go back to, remember this laser I defined in the initial conditions so that between two, let's go to the surface view. So between the times of 2 and 3 seconds, and at the right regions, this has a value of 1. So any species that evaluates to 1 at some position in space, we can use snapshot ROI to collect the values at those pixels where that species evaluates to 1. So in this case, it's at 2.4 and the laser evaluates to 1 just at those regions of space in the top membrane and the bottom membrane in this case, right? For where the photo bleaching is going to be. And so once I've defined that as the region I want to analyze, now I can do statistics in that region. So I can look at that snapshot region for, oh wait, let me go back and change this to the fluorescent species so we'll see it. And I can look at the statistics in that region. And then I actually get a perfectly reasonable FRAP curve that I can compare to my actual experimental results. Okay, so it allows you to define the regions in space that you want to collect into a particular analysis or a particular result of those species. So that's also an extremely useful way to deal with things that vary in space. And then I just want to take a second to show you another way of doing it, which is to, let's see, I have to find the right model here. Yes, okay. So this is a case where I needed to build up an actin cortex right around the edge of the cell, but the edge is, you know, oddly shaped. So in this case, I'm doing it a dictyostelium. So if I look at my geometry, the surface view here, dictyostelium is this sort of odd squat shaped species. And it's hard for me to define just a small region where I want the actin cortex to be. So you can use little tricks with kinetics. So in this case, I've created a reaction that sort of like Padmini did, you know, it comes off the membrane. So it's localized in space to the membrane. And then I'm creating a species right next to the membrane. And if I get the kinetics right, as you'll see, I can create a species that now falls off very rapidly right next to the membrane. So this is going to be a volume species. Oops. Okay. And it will, space. So it will be very high only right next to the membrane. Okay. So that's really useful. So let's say I want to say, make that value of that species be where my high actin concentration is or something, right? So one of the things I can do is I can create something in virtual cell that's called field data. So field data just basically takes a snapshot of the distribution of this species over the entire time course of the simulation. I just need the last piece, but it doesn't matter. So if I go to field data, this one is actually this one. So I can create this piece of field data from this simulation, which is the exact distribution of those molecules in space. And then I can actually use that to create a new simulation as the starting condition. Okay. Again, it's a little complex. So if you're going to do it, then talk to us or read about it in the help. So I don't want to go through the whole thing now. But you can then use that as a starting condition so you have a non-uniform distribution. You can also create field data from your original fluorescence data as well. So if, again, you have a immunofluorescent or GFP labeled molecule that you want to reproduce exactly in a spatial simulation, you can create field data from the fluorescence distribution and use that as the initial distribution in a model. So there's a number of different ways to create unique distributions. And then finally, you know, what Les mentioned early on was that you need to create a steady state, right? So if your kinetics are such that, like in Padmini's model, you'll create a non-uniform distribution based on the reactions, then the best thing to do is to run your spatial model to steady state, right? So to run it first, create the steady state. You can either then do you know, photo bleaching or initiate some reaction with an event in time, or you can save that the final distribution of those molecules as field data and use that to begin a new simulation. So those are basically the ways that we have of starting a simulation from a non-uniform distribution.
And there's a question on the chat.
Okay.
About whether it's possible in VCell to model multiple domains in a membrane and model the diffusion of the membrane proteins between these membrane domains. There's a little bit more about interactions with cytoplasmic proteins, but I think if you answer the first question. Right.
So you can, it's sometimes can be a little bit tricky for how you exactly want to do that. So in some cases, you can define, for example, diffusion coefficients that will go to 0 at the edge of a particular region. So if you start all your molecules in one region and not allow them to diffuse past the barrier, that will maintain the molecules in that region. And there are other ways of doing it in compartments. I think I might call on Boris if Boris, are you still here?
Yes, I am. If I understood this question correctly, the easiest way to do it is just to consider those membrane proteins as different species that initially defined in different domains as different species, because they have to interact with cytoplasmic proteins differently, depending on where they originated, if I understood the question correctly. But definitely you can, when you specify initial conditions, you can definitely use those Boolean conditions to specify where in the membrane the proteins are initially. Again, if I understood the question correctly, I'm not sure though.
Yeah, so you could create a membrane that is, you know, different. You could create two different membrane compartments, but it's a little bit difficult to handle the boundary conditions if they're not reaching around an entire compartment. So, so I think the easiest way to do it is what Boris says, just put your proteins in different parts of the membrane, but then you're constrained in terms of how they would diffuse laterally. So eventually they would mix, right? You'd either have to give them very slow diffusion coefficient or adjust the diffusion coefficient with a Boolean so that they would largely stay in your particular membrane domain.
Did this address the question, Yimei? I mean, maybe you can unmute, yeah?
Yeah, so my question was, I think, more about just my approach to how I should model this, and if I should have a single membrane compartment and try to define within that compartment, or just have two separate membrane compartments and just define the membrane proteins as different species to begin with.
Well, what I said is, again, so you want those proteins. Here's my question. So those proteins that are going to be defined in two different patches of the membrane, do you want them to have different diffusion coefficients or the same diffusion coefficient?
Different, if possible.
Okay, so, and you want them to interact differently with cytoplasmic proteins, right?
Yes, because if some cytoplasmic proteins, for example, are going to associate more with one membrane.
So,
Gotcha,
okay,
yeah.
so they're going to diffuse with different diffusivities, and they're going to interact with cytoplasmic proteins with different rates. So they should be different, right? So I would
Yes.
just call them differently, like protein 1 and protein 2.
Yeah. Yeah, that's what I would do as well.
Yeah.
And then they can coexist there. Yeah.
Okay, thank you.
And you can also define with Booleans as a modifier of a reaction. So if you would want a reaction to happen on one surface or another, sometimes you can just, if you know, sort of like an apical basolateral membrane situation where the apical membrane may always be a value of Z greater than 10 or something because it's up at the top of the cell. You can also define things that will modify reactions so they only occur in specific spots. Because that's the only place that would have that species if you've localized it to a region.
Okay, I'll go over some of the tutorials again, and then
Yeah,
Yeah,
and if you're building a model with us, you can always share the model with us, and we can sort of point out ways of doing it in your particular situation.
Okay, thank you very much.
Mm-hmm. Okay, go ahead.
I was going to say, if there are other questions from other people, please feel free to ask them now about any of the software.
Yes, I have a question about VCell, about spatial simulations for the diffusivity coefficient. Can it be non-isotropic first?
So could you repeat your question again? You're talking about spatial simulations, right? Or non-spatial?
Yes, in VCell, in deterministic simulations, can the diffusion coefficient depend on the direction?
Well, what we have now, no, but there is no reason why it. So you mean like you want to have dx diffusion sub x diffusion sub y diffusion sub.
Yes.
No,
Because I'm
so
working on some
what's
special
encoded now? What's encoded now seems to
it could happen that the diffusion is not the same destination.
Go ahead.
Okay. And also, is it possible to have a time-dependent diffusion coefficient, or even more diffusion coefficient that are time and space dependent and computed from, for example, reaction kinetics?
The diffusion coefficient can be function of space and time, but note that when you make it a function of time, the simulation will be slower.
And even have a diffusion coefficient be a function of one of the variables in the model.
Yeah, it can be a function of variables as well.
So you can basically type in any equation here for the diffusion coefficient. So it could be a function, it can depend on other species, things like that.
But if it depends on time explicitly or on species concentrations, then the simulations will run slower.
Right, they'll be very slow.
Well, I'm not sure.
Slower.
It depends on whether it's 2D or 3D or 1D. It might be sufficiently fast in 1D and 2D, maybe slower in 3D. Yeah.
Okay.
And you can also have advection. So you can have molecules have a velocity in one direction or another. So the diffusion coefficient can't be weighted in space, but you can you could have something that diffuses and also has a velocity in X or Y. Isn't that correct, Boris?
Yes, the virtual cell can model directed transport. So you have, for this, you have to specify Vx, Vy, Vz.
Right.
Yeah, move down on the screen and you'll see the velocity. Yeah, see velocity. So you can specify velocities for a particular species.
Right.
And that can also be a function of a variable if you really want to get fancy.
Yeah, so this can be a function rather, it can be an expression rather than just a number. Right.
Can you get curvature in there as a function of curvature?
No.
No, well, we actually struggled with somebody who was.
Well, the problem is that in deterministic simulations, the size of the particles are not explicitly resolved sizes, and therefore they can sense only what's at that point. All right.
Yeah, they're points. They don't have a volume.
I see. Okay. So you need a size of a molecule in order to for it to really sense the curvature of the membrane.
But what you can do is to make instead of gradient, you can put some kind of a concentration divided by some characteristic length. And that you can make a your expression a function of that rather than very exact gradient. If you know what the characteristic length of that gradient is, you can introduce it. But that should be a constant though.
We're getting into some very advanced, super advanced.
Right. So anyone with a little more esoteric or less esoteric questions should pipe in. Don't be shy. Most of us don't go into those realms. So we can share some simple stuff. I also need to share with you that we're going to fix a little bug that has cropped up recently in VCell. So the release version of VCell is actually going to go down for 5 or 10 minutes. So if any of you have it open at the moment, you will see it disappear. We promise it will come back. And I'm using our alpha version right now. So mine won't go away for the purposes of the talk. So
somebody asked,
and there's a general
cell population in VCell. I'm not sure exactly what you mean by cell population, but in a spatial model, you can definitely model an array of cells. You can model a tissue as, it doesn't have to be a cell-based model. Any sort of spatial model is potentially modelable.
Right, and I should point out that they don't have to be conjoined in space. So if you have 3 different cells separated by something in between them, and you define them all as the same cell type, basically the same compartment, then it'll distribute all the molecules in those 3 different distributed compartments. Okay.
Regarding populations, that can be actually a non-spatial model, which can be modeled in both capacity and virtual cell.
Yeah, I was going to say that.
So there's another question in the chat we can bring up, which is a question about the performance in spatial stochastic simulations. How does it perform as a function of cell dimension and number of species? And Boris is probably the best fit to handle that, but basically the number of mesh elements and times the number of species is how long it's going to take, basically.
Well, mesh elements do not matter because in the virtual cell actually borrows the solver from Smolden, and the version that we incorporated is where the molecules are modeled as points, as particles. So what matters is actually the number of particles of all types. And I would say just roughly, it is practical probably to run up to 500,000 of particles. I mean, you could probably tolerate waiting for the result. It's practical, but yeah, you're right. So as the time goes with the number of particles. This is spatial statistics. This is just when you really doing just stochastic simulations.
So then it wouldn't matter if I have, say, a cell which is 10 micron versus something which is half a micron.
So, I mean, stochastic simulations not, they are not dependent. You can do it without mesh, right? You're just moving in particles in space, and Smolden was optimized for. So in Smolden, there are certain tricks that that make simulations sufficiently fast. So from my own experience, 500,000 is okay, even up to 1 million probably.
But the
Of course, it will be slower than if you have just 10,000 particles.
But the nice thing about our implementation, this is really Boris's work. Boris developed a hybrid solver for spatial stochastics. So,
well, I should probably give credit also to Fei Gao and Jim Sheff. It was not just solely my work.
You were sort of head of the team, I think. Anyway, so you can, if you have something like 1 million particles, you can specify those as a concentration, and then because it's a lot of particles, you probably don't have to model it stochastically. But if there are other particles, other molecules in the system that are present in 10 or 100 copies, those can be specified stochastically. So you can do a hybrid deterministic stochastic model, and that would be a lot faster than modeling everything stochastically.
Right, yes, that's great.
Well, I should just make some kind of clarification on that. This is true what Les has said when the source of stochasticity, source of noise, comes from the molecule expressed in small numbers.
Sure.
Then you can use our hybrid solver where, say, calcium ions are modeled as deterministic, kind of, in quote marks, deterministic, and say calcium channels, their dynamics is modeled as stochastic. And of course, the noise will be conferred on deterministic variable as well.
Okay.
When to use the hybrid solver, you would create a stochastic spatial simulation, and then you can use this force continuous function for those species which you don't want to solve stochastically. Right?
I see.
You just select those, and then VCell will automatically insert as the solver the hybrid solver.
Okay.
It's sort of functionally the way you do it.